video, we are going to look at P class pump. As you remember, I had already covered briefly about P class pump, so this will be more of a revisit. And in this revisit, we are going to look at some specific examples of P class pump, for example, the sodium potassium pump. Now, let us go back and see why these P class pump are called P class. If you remember, the P comes from the phosphorylation of one of the moiety, and this phosphorylation happens in the alpha subunit. So there are two subunit, one of them is alpha subunit, and an aspirated group of this alpha subunit get phosphorylated, which is the characteristic of this pump. And then there is a beta subunit, which is having a regulatory function. So given that, now I'll look at one of the example, the sodium potassium pump, how it works and why we need it. So let us say these two yellow lines, this is the cell membrane or any membrane, cellular membrane, and outside the cell usually the sodium ion concentration is more and potassium ion concentration is less. And when you compare it to the intracellular concentration, then sodium is less inside and potassium is more. And whenever there is a voltage change across the membrane, these ions flow through their gates, if you remember, or their channels, and that creates a imbalance in the ratio of ions. So to maintain this ionic ratio, to maintain this concentration gradient, the pumps are at function. And as you see, sodium is moving from low concentration to higher concentration, which is against its gradient. So for that, energy is used here from ATP, and that is why it is a pump, and it is considered a proper molecular machine. So there are these pockets where sodium can bind, and once sodium ions bind, its structural changes cause ATP to bind and phosphorylate the aspirated group. And this phosphorylation triggers another conformational change where the sodium ion can be pushed outside. Now you can think why potassium is not binding here, and as you saw, the sites for potassium are not yet ready, or they are not active for potassium to bind. Once sodium is released outside the cell because of the conformational change, now the pockets are available for potassium binding through conformational change. And potassium ion binds from outside of the cell, and this, once the binding is complete, this again triggers another conformational change where it switches back to its previous state, and this time because of this change, the phosphorylate the phosphate group is released as inorganic phosphate into the cell, and then this again triggers another conformational change where the pocket for potassium goes through a conformational change, releasing the potassium ion into the cell and goes back to its previous state. So in this cycle, they pump sodium out and pump potassium into the cell. Okay, so let's look at another example, the calcium pump. The concept and mechanism is almost similar, but there are some differences. And this is also another P class pump, which means there will be a phosphorylation of the aspirated group. Now, what is the function of this pump? Is that, as you have already seen, this is a transporter of calcium ion, and this is present in the ER membrane so that it can transport calcium from cytosol to sarcoplasm. Sarcoplasm is the endoplasmic reticulum of muscle cells. So it is also called sarcoplasmic reticulum. And inside cytosol, the calcium concentration is less than sarcoplasmic reticulum lumen, and for that, calcium pump is required to pump more calcium into the storehouse of calcium in sarcoplasmic reticulum lumen. Now it assumes two configurations, E1 and E2. We'll study the characteristic of these things. And the E2 binds to calcium ion with less affinity, which means its binding to calcium ion is weaker as compared to E1. So here the yellow line is the SR membrane, and you see there is an aspartate group in the cytosolic side of the calcium uh, pump. Now what happens is, usually there is a ATP binding site, a domain right next to this group, this aspartate group, and when ATP binds to it, first ATP binds to this site and is cleaved, and that way in this configuration the calcium ions bind to their pockets in the calcium binding site, which is a transmembrane domain across the sarcoplasmic membrane. Now this binding activates, there is an activator domain which activates the pump by phosphorylating the aspartate group from the ATP that was bound to ATP binding site. Right, so there is, here is the ATP bound, and because of the activator domain, what we'll see is the phosphate group is transferred to aspartate group, and this is still in E1 configuration, tightly bound with the calcium. Now what happens, once phosphorylation is complete, then E1 switches to E2, and in this E2 configuration, as you see, the pump faces towards the SR lumen, sarcoplasmic lumen, and in this configuration the phosphate is still bound, but slowly, because of its low affinity to calcium, the calcium is released into the SR lumen. Okay, calcium moves from its pocket to the SR lumen because of the configurational change of the pocket itself, from the configurational change of the entire protein to E2. And in this situation, we have the pump in E2 state with calcium gone, but phosphate group still bound to aspartic acid group. And what happens is that triggers the dephosphorylation, and inorganic phosphate is released to the cytoplasmic side, and this switches back the E2 to E1, where it is again facing towards the cytoplasmic side. After looking at the calcium pump, let us look at something called secondary active transport. Now, as you saw, in sodium potassium pump or calcium pump, energy is used to transport something. However, in secondary active transport, the molecule is transported across the membrane by using energy obtained from concentration flow of a molecule across its concentration gradient. So that makes the transport a passive one for the molecule in concern, but there is energy consumed for transporting the other molecule, which is to be maintained in a concentration gradient across the membrane. Now we'll look at some of the feature of this one and an example also. So one species of molecule is getting transported along with the movement of another species across the membrane. Now, as you see, the two different molecules, two different species are moving across the membrane. It also raises possibility in the directionality of their movement. So according to this directionality, it can be symport, where both of them are moving in the same direction. Or it can also be antipode. In antipode, as you would have already thought, they are moving in opposite direction to each other. Now we'll look at one example, which is the transport of glucose across the membrane, which happens with the help of sodium ion transport. So here, let us say we have a cell membrane. So this is the lipid bilayer of the cell. Let's consider some. So let's consider one of the sodium potassium channel, sorry, pump, which is pumping out sodium to maintain a concentration gradient of high sodium outside the cell, right? Such that sodium can flow from outside of cell to into the cell, and then we have glucose getting transported in this symporter transport protein using the same energy generated from the transport of sodium across its concentration gradient. However, why is it secondary active transport? Where is the active coming from? Here we have a sodium potassium pump, and this one is actively sending sodium out of the cell to maintain this concentration gradient that glucose just used to enter the cell. Without this pump on the right, the concentration gradient will not be established, which cannot be, in that case, the sodium and glucose transporter will not be able to transport glucose. So here glucose is going through a passive movement, while sodium pump is acting through an active transport method. Now this mechanism is used in a lot of physiological conditions. For example, pushing glucose into tissues which already contain a lot of glucose, like muscles or liver cells. So we'll look at this physiological regulation in later video. So here we have a sodium potassium pump, and as you can see, here we have this region is the region outside the cell. This is the transmembrane domain, and here we have the inside the cell part, inside the cytoplasm. If you look closely, these purple balls, these two are the potassium ion, and as you can see, if I take you little closer, the green ones you see there, those are, one of them is a magnesium ion, but the one I want to show you is just wait. If I keep my cursor here, you can see it writes Ca2005K, which means potassium for K, and the MF4, which is magnesium tetrafluoride. It mimics the phosphate group and can inhibit any protein dependent on ATP like moiety. So this pump is being kept stuck in this state so that its structure can be determined, and we have a normal magnesium ion which helps the protein function. And from here you can appreciate how complex this protein is from all the alpha folded barrels.